Hello, dear students. This is Lázaro Pino, your chemistry professor. Today we are seeing proteins. The amino acids are the building blocks of the proteins. Also, we are going to see today the functions and the structure of proteins. Proteins come from the Greek proteus, that represents something like primary or of first importance. They are biomolecules also, and they consist of polypeptides joined by peptide bonds between the amino and the amino and the carboxylic groups in the amino acid. Proteins perform a number of important functions in our bodies. For example, they can be enzymes. They can be part of a structure of our body, or also we can see them in the diet, and they have several importance in the cell. For example, in the immune responses, in the cell adhesion, and in the cell cycle. Here you can see a protein. The protein is made of carbon, hydrogen, nitrogen, and also you can observe sometimes between the molecule hydrogen bonds. The amino acids are made of two main groups, an amino group and a carboxylic group that we have studied in organic chemistry. Both are together bonded to a central carbon, and this carbon is bonded to a hydrogen. This part of the molecules is the same for every single amino acid. What makes the change? The change is made by the side change that can be different, and according with this you have 20 natural occurring amino acids to build proteins. The linear sequence of amino acid in protein is dictated by the sequence of the nucleotides in an organism's genetic code. Here you have the amino acids can be divided into non-essential like alanine, asparagine, aspartate, glutamate, and serine, and conditionally essential that are arginine, cysteine, glutamine, glycine, proline, and tyrosine. The essential ones are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. These amino acids are called alpha amino acids because the amino groups are attached to first carbon in the change and connected to a carboxylic carbon. Here you have the classification of the amino acid. According with the side chains, they can be basic or polar, they can be acidic polar, or they can be neutral non-polar. The stereochemistry of the amino acid says that they can have L and D isomers according with the position on the spades. These structures are known as enantiomeric forms, as we studied in carbohydrates. The sweeter ions are because amino acid contain both an acidic and a basic functional group in the same amino acid. So, an internal acid-base reaction occurs, forming an ion with both a positive and a negative charge called sweeter ion, as you can see here. Then, according with the pH, there will be changes in the structure of the amino acid. The peptide formation is when an amino acid is bonded to another amino acid. The bond that is made between two amino acids is known as peptide linkage. Remember that in carbohydrates the name was um, uh, glycosidic bond. Here you have an important amino acid. This amino acid is vasopressin. This is the formula, a little complicated, yeah. But the importance of this amino acid that is produced by the posterior pituitary is very important because they can have two mechanisms: the blood vessel constriction and also kidney fluid reabsorption. According with this, the arterial pressure can be increased. Other example of peptide and proteins hormones are the ones that you can see here in this table. For example, the HGH, the human growth hormone produced by the pituitary, is a very important one because it has a very important action in the bone growth. Just to give you some examples. You can stop the video at this time and you can go over different ones to see the important peptides and protein hormones. The characteristic of proteins depends on the size. As you can see, for example, in glucose, the molar mass is 180, while hemoglobin, that is an amino acid, the molar mass is 65,000. Then, as you can see, proteins are micromolecules, bigger than any other one that we have studied before. You have here for insulin that the molecular weight is 6,000, and for example, for gamma globulin is 176,000. Myosin is 100,000. Then the function of the proteins is very important. I say protein at the beginning is primary, is essential. Well, it is. They are, because they can be catalyst, they can be a structure, they can be a storage, they can be protection, they can be regulatory function. Then here we have the hormones. They can be used in the nerve impulse transmission, and they also can be used in the movement function. I strongly recommend you to stop in here and read carefully all these importance of the proteins. Here also we have a very important one that I put separated because it's a transport function. They can be transporter. For example, some proteins bind small molecules or ions and transport them through the body. For example, the serum albumin is a blood protein that carries fatty acids between fat, adipose tissue, and other organs. Hemoglobin carries oxygen, everybody knows that, to the protein, to the tissues, and from the lungs. And transferrin is a carrier of iron in blood plasma. A typical human cell contains 9,000 different proteins. The human body at all contains 100,000 different proteins. Here is a very important thing. That is the structure of protein. The primary structure of protein represents the long chain of the different amino acids bonded by peptide bonds. The insulin, the human insulin, has this kind of a structure, but the secondary structure can give you an alpha helix or a better pleated sheet, and it's because between different strands of the protein we have several hydrogen bonds that makes this a structure like a helix or this in a plane, as you can see. The tertiary structure of proteins is because proteins in the liquids are rounded like this one, like the one that you can see in this part. The tertiary structure of proteins depends on different kinds of bonds, not only hydrogen bonds as we have seen in the secondary one, but also we have a hydrophobic interaction, we have salt bridges, we have also disulfide bridges. And the quaternary one is when you have several tertiary structures joined together to make a globular structure like this one. Hemoglobin is an example of this. Let me give you an important thing about hemoglobin. When a person suffers from sickle cell anemia, it's because one amino acid difference in the sixth position of hemoglobin, it changed the glutamate by valine. And then the shape of hemoglobin is different because of this shape that is no globular. We have seen before, they can block capillaries, and of course they are rapidly destroyed. And then the lack of hemoglobin produce anemia. Also, when they go into a small capillaries, they produce a very strong pain. Substances that can destroy the protein. Well, here you can see the heat and the ultraviolet light. Then the temperature is very important for the effect of proteins. The organic solvents, for example ethanol and other miscible with water, disrupt the hydrogen bonds. Also, strong acid and bases can disrupt hydrogen bonds and the ionic interactions. Detergents and heavy metals too. Heavy metals like mercury, silver, and lead can form thiol groups and precipitate the proteins, and insoluble heavy metal salts form. Here in this table you can see different types of proteins. Here we have collagen. Here we can see the cytochrome, the purine, the aspartame, insulin. Oh, there are so many examples of proteins. That's why I say proteins are maybe the most important micromolecules in our bodies. Well, to the end, I left one important action, the enzymes. Enzymes are catalysts that try to speed up the reaction. An enzyme is made of an active site where the substrates are binded, and when the substrates are binding in here, then a weak bond that join them together is broken down and produce the products, and beside that the enzyme is ready to be used once again. We have different types of enzyme. Amylase, that is used for the digestion of carbohydrates and is in our mouth. Lipase, that is used in the digestion of fats, and we can find it in different parts of our digestive system. Pectin for proteins, trypsin for protein, lactase for dairy, and they are in the small intestine. Well, but there are several important factors that can affect the action of the enzyme. They need a very specific temperature that in our body coincides with 37 that is the body temperature, to be active. Below this is not activated, and after that is destroyed or is spoiled. The pH is also important, as we have seen before. That is an optimum pH. Below is not activated, after this pH is destroyed. The concentration of the substrate is also important, because this reaction increases until certain amount of substrate that cannot be digested or can be used by an enzyme to be divided into a small units that can be used in our body. Well, that's it for today. As I told you before, proteins are very, very important.